Welcome student. So in this period we are going to study about the TS of anther. Actually in the last period I had told you that anther is nothing but it is a group of parenchymatous tissue. So when the maturation starts, some of the cells of this parenchyma tissue known as hypodermal cells they get transform म्हणजे त्याचे स्वरूप बदलते. So they are transformed into archisporial cells. They are transformed into archisporial cells, and later on these archisporial cells divides into two types: the outer cells and inner cells. So the outer cells actually they develop into parietal cells, and the inner cells they develop into sporogenous tissue. Now these parietal cells later on develops into anther wall. So wall of the anther it is produced by parietal cells, and the sporogenous tissue, no doubt it is a tissue and it is made up of sporogenous cells. So each sporogenous cell later on gives rise to a tetrad of microspore. In short, we can say each sporogenous cell it produces a tetrad of microspore, and you know that microspore means pollen grains. So see here, suppose this is a single cell of sporogenous, and this is a single sporogenous cell. Later on it divides and it gives rise to a tetrad. You know tetra means 4. So these are 4 pollen grains. So 4 pollen grains or 4 microspores are produced in the single sporogenous tissue. Now we will see about the anther wall. So see here, the wall of the anther it is composed of, number 1, the outermost layer is called as epidermis. So epidermis is outermost layer and it is protective layer. It is composed of tubular cells. Now just inner to the epidermis there lies epithelium. Epithelium is there. Third layer, that is inner to the epithelium, that is known as middle layer. Actually it is 1 to 2 layered structure, and then fourth one is tapetum. So tapetum layer it is inner to the middle layer, and this is the important structure in the wall of the anther because tapetum is nutritive in function. So you know nutrition, that is portion. So this tapetum it provides nourishment to these developing pollen grains. त्यांना तो अन्न पुरवण्याचं काम करतो. This is the tapetum, and thus the tapetum encloses these sporogenous tissue. So sporogenous tissue it is present within the tapetum. So if we have to draw the structure of TS of anther, how we can draw the structure? See here like this. Actually this diagram is given in the textbook. So you can practice at home like this. Structure is there. Here there is a connective. This is anther, actually 2 lobe anther, and in the last period I told you that Each lobe contains 2 pollen sacs. So see here 3 and 4. 4 pollen sacs are there. Now we will see, as this is the anther wall. This is the anther wall. Outermost layer, just now I had told you that epidermis. So see here, this is epidermis, somewhat tubular cells. So I will draw only short part. Beneath the epidermis, so this is number 1, that is epidermis, and number 2 epithelium. So beneath or below the epidermis, see here like this, there lies the second layer, that is called as epithelium. Okay, now again below that epithelium there is the structure that is known as, here third one is middle layer. Okay, and the last one is tapetum. So here, see here, this is the tapetum. This layer is called as tapetum, like this. This is the fourth one. Okay, and this tapetum encloses the sporogenous tissue. Now you can see here, inside this tapetum encloses the structure that is known as, here, sporogenous tissue. So you can write here number 1 epidermis, second one endothecium, third one middle layer, and the fourth one is tapetum. Fourth one is tapetum, and here inner, these are the sporogenous tissue which later on gives rise to microspores. So the next part is now microsporogenesis. Microsporogenesis. Here microspore, microsporo or microspore, genesis, that is formation. So here microspore mother cells, microspore mother cells in short they are written as MMC. These microspore mother cells, no doubt these are produced by the sporogenous tissue present here. So microspore mother cells, they are no doubt diploid in nature. They undergo meiosis type of cell division. Meiosis type of cell division, you know that it is a reduction division, and it produces the tetrad of microspores. Tetrad of microspores which are haploid. And you know that haploid, we can write here n number of chromosome, and diploid means having 2n number of the chromosomes. So this is called as microsporogenesis. In short, formation of microspores from the microspore mother cells by the process of meiosis is called as microsporogenesis. Okay, now we will see the structure of microspore. Structure of microspore. So here microspore you can write as a single pollen grain. Okay, see here each microspore is number 1 unicellular, uninucleate, it is non-motile, it is double walled, etc. Now how you can draw the structure of microspore? See here, so this is microspore, nucleus is there, cytoplasm is there, like this. Suppose you can draw the double wall of the Microspore. See here, this is the microspore. See here, the outer wall is called as exine. The inner wall is called as intine. This is the cytoplasm. This is the nucleus present in the cytoplasm. See here, what is difference between exine and intine? Actually, the exine, as it is double walled, outer layer is exine, inner layer is intine. This exine, it is tough, intine is somewhat thin. And here this exine, it is not continuous, it shows some areas known as germ pores. Some thin areas are there actually. It is not continuous, it shows some thin areas, and that thin areas are called as germ pores. And the intine, no doubt it is thin and it is continuous actually. Now, through this germ pore, during the germination of the pollen grain, pollen tube comes out. So, pollen tube comes out through the germ pore. Exine is made up of the chemical known as sporopollenin. They are made up of sporopollenin, whereas intine is made up of cellulose and pectin. Cellulose and pectin. Okay. So, this is about the exine, intine, the double walls of the microspore. Okay. Now, the next one is how male gametophyte is formed. So, that is called as development of male gametophyte. Development of male gametophyte. See here, one thing I have forgotten to tell you, that this double wall structure of the pollen grain, it is also called as sporoderm. That double wall structure is also called as sporoderm. Okay. Now we will see the development of male gametophyte. See here, once you know the structure of the microspore, we will see now the next. No doubt, male gametophyte develops from this unicellular, uninucleate, non-motile, double-walled microspore. And here, this microspore actually also known as pollen grain, it undergoes the cell division known as mitosis. So, first of all, mitosis 1 takes place in this pollen grain, and it gets divided into a two-celled structure like this. This is the two-celled microspore. Two-celled microspore. Now see here, two cells which are unequal. So the larger cell, it is called as vegetative cell. So vegetative cell is larger, and the smaller cell, this is the smaller cell, it is called as generative cell. Generative cell. See here, generative cell and vegetative cell. Now, this microspore, two-celled microspore, again undergo second type cell division, that is mitosis 2. Actually, mitosis 2 occurs only in the generative cell, only in the generative cell. Now see here, actually the generative cell, it floats, है ना, generative cell, it floats in the cytoplasm of the vegetative cell because it is larger cell. Now what will be the structure? See here, like this. This is the vegetative cell is there. Okay. Now here it divides into two. No doubt exine is there like this. So such type of structure is formed, and here these two newly formed cell, they are called as two male gametes. So each generative cell gives rise to two male gametes like this. Okay. Now after that, the intine, the inner layer that is called as intine, due to pressure the intine comes out. So here suppose we can write here, such here germ pore is there, hai na? So through the germ pore the intine comes out like this, the outer exine. Now like this, here this structure is called as pollen tube. So pollen tube is nothing but it is the extension of the intine, hai na? And here, now see here, this is the nucleus of the vegetative cell. See here, this vegetative cell, it is also called as tube cell. It is also called as tube cell. So this nucleus of the tube cell, it also enters into the pollen tube. The cytoplasm of this tube cell or vegetative cell, it also enters into the pollen tube. Similarly, these two male gametes, they also enter into the pollen tube. Now this structure of the microspore, this structure is called as male. You know, this is the symbol of male, hai na? So this is the structure of male gametophyte. Okay. So what is male gametophyte? Male gametophyte is nothing but it is the pollen grain with pollen tube in which there are two male gametes. So these are two male gametes. Two male gametes in the pollen tube. This is the nucleus of vegetative cell, or tube cell. This, and cytoplasm is also. So this total structure is called as male gametophyte. So here we complete development of male gametophyte. Thus male gametophyte develops from a single microspore or single pollen grain. And the last important point is that you know about the pollination. During pollination, pollen grains are transferred from the anther to the stigma of the receptive flower. So see here, this stage is very important. That is two-celled microspore stage. So at this stage generally, so at, we can write here two-celled, at two-celled stage of microspore. So at this stage they are released. They are released from the anther, or we can say they are shed, released or shed from the anther. Okay, students. So I think all of you understand here the steers of anther, structure of microspore, and the third unit is development of male gametophyte. Okay. So okay students, study well. If you have any problem, please ask me. Okay.